Okay. All right, we're live. Okay, let's see. I guess we'll just go through the key. Yeah, this was a lot of just definition stuff again. Oh my gosh. Okay. All right. So number one, so the mRNA sequences, rRNA and tRNA molecules evolved in initiation of translation. So the mRNA sequences are the Shine del Garno and the start codon. The RNA molecule is the 16S RNA, and the tRNA molecule is the initiator tRNA.
Are you just reading off the key?
Yeah. Okay. Is that okay?
Yeah, that's good.
We can stop and go over anything if anyone has questions about them.
No, it was fine. I was just curious.
Okay. And what proteins are involved in translation initiation in E. coli? So that's the IF3 and IF2, which are the two initiation factors. What mRNA sequences and RNA molecules are involved in elongation. So the mRNA sequences each sense codon. And then the RNA molecules are 16S rRNA and 23S rRNA. And then the proteins involved in elongation are EF2 and EFG. And then the mRNA sequences and proteins involved in termination of translation. So the mRNA sequence are the stop codons, so 5' UAA 3', 5' UAG 3', and 5' UGA 3'. And then the proteins are RF1 and RF2.
Okay.
Okay. So then the next one asks about the different tRNA anticodons. So for 5' UUU 3', it would be 3' AAG 5', 3' AAA 5', 3' AAU 5', and 3' AAI 5'. What's the I? You guys know what the I is?
In the PowerPoint, it shows the little key. Did you see that? So in the PowerPoint, it tells you when you have that specific thing being tested for, that the 3 results would be that, and then it gives you I as one of the results.
Okay.
It's a little box. Yeah. It's like the wobble rules. It has the I on it.
Okay. Sounds good. Okay. That makes sense. I haven't looked at the PowerPoint.
Yeah, the PowerPoint usually never works for me.
Yeah, usually everything's just in the, yeah. Okay, then for B, for the 5' GCG 3', would be 3' CGC 5', 3' CGA 5', and 3' CGU 5'. And then the tRNA anticodon has 5' IAA 3'. So the mRNA could have 5' UUU 3', 5' UUC 3', and 5' UUA 3'. And then the tRNA anticodon has a sequence 5' UAC 3'. So then the mRNA could have the 5' GAU 3', 5' GAC 3', and the 5' GAG 3'.
For that one, I added an extra one. I had 3' AAG 5', but that's not part of one of them. Do you know why it wouldn't be one of them?
For the last one?
Yeah.
What was the question? Sorry.
So I added one sequence to it, which is a 3' AAG 5', but it's not included. Because if you have the A as a tRNA anticodon, you're supposed to have U, C, G, and A. Well, it's sometimes A because it's in parentheses.
Yeah, I don't know. That's kind of weird.
Okay.
Just forgot it.
Yeah. Okay. So the mRNA E. coli molecule has the following sequence. And so it's a really long sequence. Do you guys want me to read it out or no?
I'm good.
How long is it? I don't think so.
It's
I
like
haven't looked at it yet, but I think.
23 bases or something.
Okay, I think.
So identify the ribosome binding site, start codon, and stop codon. So the ribosome binding site is going to be the Shine-Delgarno sequence. So that's the AGGU. And then the start codon would be the AUG. And then the stop codon in this one is UAA. And then the amino acid sequence is F-methionine, glycine, aspartame, lysine, tyrosine, glycine, glutamine, I believe. And then if the underlying G is deleted, it would be F-methionine, alanine, isoleucine, aspartame, methionine. I don't remember this one. I'm just going to say ALA, serine, and lysine. And then if the 3-fold bases are deleted, then it would be F-methionine, glycine, lysine, tyrosine, glycine, glutamine. And then if you, the amino acid sequence, if the base in the italics is changed to an A, it would be F-methionine, glycine, aspartame, lysine.
How do you get those?
So I just looked on the, there was a chart in the PowerPoint, and you just go by you just count off the three base pairs into each codon, and then it will tell you which amino acid is associated with it.
Oh, is it the yellow one?
Yeah.
Okay. Gotcha. That would take forever.
Yeah, it does.
I didn't do that one because I wasn't sure.
Okay, for number 4, what is it meant by the term charged tRNA? So it's a tRNA that is covalently linked to amino acid. That is to the 3' end of the tRNA. How many ATP molecules are consumed to charge a tRNA molecule? And that's 1 ATP. The initiation step of translation incorporates one amino acid into a polypeptide chain. How many GTP molecules are consumed during the initiation step of translation? It consumes 2 GTP molecules, one by IF3, another by IF2. The elongation step of translation involves repeated cycles, with each cycle incorporating one amino acid into the polypeptide chain. How many GTP molecules are consumed during each of these elongation cycles? So the elongation step of translation consumes 2 GTP molecules. 1 GTP is consumed by EF2 and the other one by EFG. And how many GTP molecules are consumed during the termination step of translation? And so that 1 GTP molecule is consumed during that termination step, and then that is used by release factor to cleave the bond between the polypeptide chain and tRNA molecule in the P site. Any questions on any of those?
I don't think so.
Okay. All right, so shown below is the amino acid sequence of 3 mutant proteins that are produced by a single nucleotide change in the mRNA. Here, wait a second, my dog's asking to go out. Okay. So they have like the first, so the wild type, yeah, so the first one is the wild type. The second one stops after 2 amino acids. The third one just has like, let's see, it has a change in the tyrosine to histine. And then the third one, or the fourth one, totally changes all of the amino acids. Do you guys want me to read them out, or is that okay?
You're good.
Okay. So one possible mRNA sequence for the wild type protein, so that could be 5' AUG, UGG, UAU, CGU, GGU, UCU, CCU, ACU. 3'. Okay. Sorry, wait 1 second. Okay. All right. So for the first, so it wants to talk about this specific nucleotide change in the mRNA that leads to the synthesis of each mutant protein. So in mutant 1, a change from U to G in the third codon would generate the protein because it makes a stop codon. And then mutant 2, a change from U to C in the third codon generates the mutant C protein. That's just that entire sequencing. And then mutant 3, a deletion of the second G nucleotide in the second codon generates the mutant 3 protein. The second mutation is a frame shift. That's the only one that I didn't get. I had a hard time figuring out that one.
I didn't get any of them.
Yeah, this one was definitely, it's hard because they're all like, there are like a couple codons for each amino acid, so you kind of have to like look at each one and figure it out. It takes a long time.
Yeah.
Do you get how to do it though?
No, this is the one I didn't understand.
Okay. So this one, you just have to like match the amino acids that they give you to like the different codons. And then for like the different mutants, so you look at the wild type and then you look at like, so the first mutant, you would look to see like, where's the change in the amino acids? And you see that it like stops early. And so you know that there's a stop codon before there's supposed to be. And so that would be like in the tyrosine one that changed. Does that make sense? Like there was like a wrong base put in.
Okay.
And so
Did you use
stopped instead of
the genetic code slide again for that?
yeah.
Okay.
So
It's lots of info that I'm missing out on.
yeah.
Okay.
But does that make sense?
Yeah, a little bit.
Yeah.
And then you would do like the same thing for the other ones, just like find where the change is and then get it out.
Okay.
There.
I guess I didn't start that way.
Okay. Then part C, the mRNA that produces the wild type protein should have 9 codons. What is the functional role of the 9th codon? And so it's the 9th codon is a stop codon, and that's what's recognized by the release factor and terminates translation. The stop codons would then be 5' UAA 3', 5' UAG 3', and 5' UGA 3'. The mRNA molecule that produces the wild type protein contains a 5' untranslated region upstream of the codon. What sequence is contained in the 5' UTR? what is the purpose of the sequence. So that's the Shine-Delgarno sequence. It will form hydrogen bonds with the 16S rRNA, which allows the ribosome to bind to the mRNA. Okay, so that's all of it.
Yep.
Is everyone good with it?
I think so. I don't know.
Adam just looks like
Sure.
he's still like unsure.
Yeah, I don't know. I'm just really tired, so I'm just. I don't know. I mean, I tried to answer them all, but I didn't get a lot right. So.
Yeah, I didn't get a lot right either.
The applying genetics isn't too bad. It didn't take me too long.
I'll have to figure out some way to get the PowerPoint to work, because every time I try and download it, it just like won't open, and it'll have like errors and stuff.
That's weird.
Yeah. Yeah. But
All right, I'll
figure
send
it
you
out.
the link.
Okay.
Okay, bye.